Hi, I'm Christian Mar Obainting.
And I'm Alan Julius Razal.
Today, we will show you how to extract DNA from a banana to investigate how it looks and feels.
This procedure is similar to what scientists have to do before they can use the information contained in the DNA. The scientists are able to separate DNA from the unwanted substances of the cell gently enough so that the DNA does not break up.
The salt allows the DNA strands to come together and precipitate out of the alcohol solution. The dishwashing liquid breaks down the cell membrane by dissolving the fatty molecules and proteins of the cell and disrupting the bonds that hold the cell membrane together.
The dishwashing liquids then holds to these pieces, allowing them to filter out of the solution by the coffee filter while leaving the cell's DNA in the liquid that breaks to the filter. So the materials needed in performing this experiment are the following.
Of course, banana, graduated cylinder, mini graduated cylinder,
Isopropyl alcohol, dishwashing liquid, salt, a cup of water, funnel, tape,
spatula, teaspoon, tissue, and plastic cellophane instead of resealable plastic. Other materials: pair of scissor, strainer, and stirrer. The first step is to peel the banana. After peeling off the banana, place it inside the plastic cellophane and seal it with tape. Mash the banana for about a minute until all the lumps are gone and it almost looks like pudding. After a minute, it will look like this. Second step is to make a salt and water solution. 50 ml water and 1 teaspoon of salt. Use a stirrer to mix the salt and water.
The third step is to pour the salt water mixture into the bag. Close the bag and very gently squeeze and move the salt water mixture and banana mash together. Do this for 30 to 45 seconds. After 30 to 45 seconds, it will look like this. The fourth step is to add 1/2 teaspoon of dishwashing liquid. into the bag. And gently mix together. The fifth step is to place the coffee filter in a graduated cylinder, securing the top of the filter around the tip of the cylinder. The sixth step is to pour the mixture into the filter and let it sit until all the liquids drip down into the graduated cylinder.
You can also use the strainer to easily get the extract before you put it into the coffee filter.
The seventh step is to remove and discard the filter. Eighth step, tilt the glass and slowly add alcohol down the side of the graduated cylinder.
Last, we observe our product, the DNA in a graduated cylinder. After the alcohol is set up, wait for 8 minutes. You may see some bubbles and cloudy materials moving around in the alcohol. This is the DNA pieces clumping together. The DNA of banana appeared white and formed a clump made of string-like strands. To summarize the main steps involved in extracting DNA from bananas, we crushed the bananas to help release the DNA. We made a solution: water, dishwashing soap, and salt to free the DNA from other components. The dishwashing soap breaks apart the cellular and nuclear membranes, and it releases the DNA. The salt helps the DNA strands come together. We used coffee filters to remove large particles, and we used alcohol to precipitate the DNA. DNA is not soluble in alcohol.